to pray for us as we begin our session. So if you would want to volunteer, kindly indicate by a show of your hand so that we can pray. Yes, Chiveru Peter, please proceed.
Let's humble ourselves for a word of prayer. Heavenly Father, we want to thank you for our lives. We want to thank you for protecting us. We pray, Jesus, Father, King of Glory, that as we study, may we understand each and everything that we are going to be taught. And we pray, Lord, Father, King of Glory, that we remember it as we do our final exam. We give you the glory and the honor. In Jesus' mighty name we have prayed. Amen.
I want to believe that Chiveru is done. Amen. Okay, so let's start. I'm aware that many of us are different levels of coverage, but I opted to pick a few questions in areas I feel that most of us have covered, and I want them to be the basis of our discussion. So I have a question I have picked from some of the papers that have been done recently, and I feel that it would serve as a good question for us to begin our revision. So here is the question. We are going to read it out together, of course, quietly from your ends, and then we see how to go about this question. So this question goes as follows. The table below shows the amounts of substances in milligrams from fluids obtained from the nephron parts labeled P, Q, and R. So the nephron has parts P, Q, and R. So we are being told to study the table carefully and use the information in the table to answer the questions that are attached. So we see 6 columns. The first column shows fluid components. These are water, protein, urea, glucose, and sodium ions. Then we also see the amount of substances in milligrams per milliliter of the plasma from part labeled P. So we see the amount of the density of water. We can also see the density of proteins, the density of urea, glucose, and sodium ions. Then percentage concentration of the same substances from the part labeled P, okay? And then we also see the amount of substances, again, the density in the glomerular filtrate from part labeled Q. And then we also see the same substances, how they vary when it comes to the urine. And this urine was picked from part labeled R. Then we have been provided with space to calculate the percentage concentration of substances in the urine in part labeled R. We shall see how we do that. So I hope we are all familiar with this kind of question. This is a question in one of the topics we are expected to have covered by this time. This is a topic of excretion. Excretion. Maybe very quickly, can we have someone tell us what this topic of excretion is all about. What do we mean by excretion? Feel free to raise your hand. Yes, Camila?
Excretion is the removal of waste products from the body by the process of metabolism, is it?
Okay, well tried, Camila. Camilas. Can someone else add on or subtract from what Camilas has shared with us? Okay. I see Tracy.
Excretion is a removal of metabolic wastes from the body.
Okay, thank you. So excretion is the removal of metabolic waste products, okay, from the body. So during metabolism, there are certain useful products that are generated, and in addition, we have waste products, and we all know that waste products are toxic, so they should be removed, or else the organism will suffer from some illness, or it could eventually also die. So that's what excretion is all about. Yes, I see Camila's hand has stayed up. Do you want to ask something? Okay, kindly after responding, you put down your hand. So that's what this whole thing is about. And we have taken a small section in the human body, that is the nephron. So where do we find the nephron? Which part of the human body contains a nephron? You see, when we are attempting questions, it's important to first understand what the whole question is about before even venture into attempting it. Yeah, someone who goes by Galaxy A30, please respond.
Kidney.
If your device is Galaxy A30, please respond. Okay, let me take someone else. Peter?
So I think in the
Say,
kidney.
say.
Yes, Peter? The stage is yours.
It's in the kidney.
Yes, so the nephron is found in the kidney. So the nephron is the functional unit of the kidney. So your kidney contains millions of nephrons, and each of them serves as a microfilter, okay? So microfilters that are there to filter the blood that comes through so that the waste materials of metabolism are removed before the blood can be allowed back into the general circulation. So in this question, we are being told that the nephron has three parts, P, Q, and R, and we are expected to appreciate the different parts of the nephron and what takes place in those different parts of the nephron. So you'll notice that some fluids were taken out from part labeled Q, which we don't know yet, but we can find out. And then a part labeled Q, and then another part labeled R. So we are supposed to use this information and be able to make an intelligent guess on where these fluids were picked from, based on the knowledge that we have about how the nephron operates. Okay. Yeah, so let us first begin with the first part of the question. We are being asked, so I hope we've studied the information. We can see that some of the contents vary. Some of them are increasing while others are decreasing, while others are altogether absent from the, as we move along the different parts of the nephron.
Yes.
So let's look at the question that proceeds.
Let's check microphone 1, 2.
Oops.
Yes, check microphone 1, 2.
So we have the first question. We are required to complete the table by filling in the percentage composition of each component in the urine. If the urine has a density of 1009 milligrams per milliliter, I think at one point we have gone to the hospital and we have been able to take a urine test. And when we do a urine test, what we call urinalysis, they check for a number of things. And one of the things is what we call the specific density. And so here we've been given the density of the urine, and we have been given also the density of other components. So how can we use the density of urine to establish the density of these other components as we can see them here? For example, how would we establish the density of water in the urine? Okay. Knowing the density of urine itself. So what is the percentage concentration? May I see a show of hands? May I see by show of hands anyone who wants to attempt this? Master Care Christian High School, do we have any participants who are willing to take on this question?
Yes, we have.
Yeah, please respond.
We are still calculating.
You
We are
could tell
still
us the
calculating.
formula, because I've already calculated it somewhere among the answers. I just want to know whether we have the right approach to the question.
Okay. Okay, sir. Okay, so
anyone else who is ready? Okay, Daniela.
For the first one, I would say 959 divided by 1009 times 100.
Okay, thank you, Daniela. Yeah, so that is how we go about that. We are going to get the contribution of that substance or component to the urine, divide by the overall density of the urine, then multiply by 100 so that we can get now the concentration, okay? So that we can get the concentration. So if we did that, we should be able to get the following values. I hope we have all understood how we have arrived at that. If there's still some unclear thing, please do not hesitate to bring it to my attention. Okay, silence seems to mean that we're on the same page.
Yes.
Okay.
Yes.
Please pardon.
So this is how we calculate it, like Daniela said. So in the last column, we had 559 milligrams per milliliter. So we divide it by the concentration, of the urine itself, and then we multiply by 100. So we should get a 95.0%. And when we do for protein, you notice that in the urine, there were no proteins. So we 0.0 divide by the concentration of the urine, and then we get 0%. Then for urea, we get 2%. For glucose, we are getting a 0.05%. And for the sodium ions, we are getting a 0.35%. Okay, so that is how we go about that calculation. So
Okay,
if we
thank you.
look back at our question here, before we go to the next question, we are required to appreciate the variation in these components in part P, part Q, and part R. Okay, so how can this variation help us know from which part they were taken? So in other words, what is part P, what's part Q, and part R? Because they're asking us in question 2, identify the parts of the nephron labeled by P, Q, and R. Actually, I missed the R here. It's supposed to be P, Q, and R. So what parts are those from which we took this?
P is the glomerulus, Q is the Bowman's capsule, and R is the ureter.
Okay, do we have any other submissions? So she says P is glomerulus, then R, you may want to repeat.
I said P is the glomerulus, Q is the Bowman's capsule, and R is the ureter.
Okay. I think some of your answers are correct, but others are not correct. So the P is not the glomerulus. Okay. However, the Q is the Bowman's capsule, and R is not the ureter. Okay. Because the ureter is not part of the nephron. The ureter is outside the kidney. Okay, so that makes P and R are not correct. So can we have someone who wants to tell us what the P and R are? Maybe I can show the information once more so that we can appreciate what P and R might be.
I'm around.
I expect as many of us to be participating. I'm seeing only a few persistent.
Can I correct
So
myself?
Benjamin, if you want to say something, I suggest you raise your hand and you only unmute when you have been picked. Let's have Benjamin contributing.
Yes, teacher, I think P is the afferent arteriole.
Afferent arteriole. Okay, that's correct. And what would R be? What is R? What is R? Winnie?
Distal convoluted tubule.
Distal convoluted tubule? Not quite. Jocelyn?
Loop of Henle.
Loop of Henle, not quite. Another one, another try. Camillus?
Collecting duct.
Yeah, it could be the collecting duct. Okay. All right, so can we understand why those answers are correct?
Yeah, we want to
Now
know. We want
remember
to know why
P,
the answers are correct.
yes, I see there's some stuff in the chat. Maybe, okay, sorry, those who are not looked at the chat. Okay, I see some correct answers
Yeah.
in the chat. Okay, now, why is P the afferent arteriole? Afferent arteriole. Remember, we have two arterioles, the afferent, starting with E, or efferent, and then A, that starts with A, afferent. So how do we tell them apart? How do we tell them apart? Maybe I can, okay, let's first hear someone say something, then I'll show you the image. We have Galaxy A30. I hope this time you talk. Yes, Galaxy A30. Can you unmute yourself so that you can talk? A30?
Master K, as we are talking,
I think because the
and so
afferent,
say, and
the
so say,
afferent is a bit,
Nakaona kuogira.
the afferent is bigger than the inferent because of the pressure it takes.
The afferent is bigger than the efferent. That is a true observation, but in this information we have at hand, can we really know that when we look at this data? Is the data enough to help us know that the one is bigger than the other? Tracy?
I personally think it's because there is protein in the afferent arteriole. Because if we're to look at the difference, if the urine was to go, if the urine was to go down, there would be no protein. So the only place that has protein would be part P, which is the afferent arteriole, I think.
Okay, that's very beautiful. Yeah, it's only in the afferent where we see protein. Okay, now after the afferent, then we have the glomerulus. Then we have the efferent. So at the point of the afferent, we have proteins. Then in the glomerulus, we also have proteins. But when we go to Q, in Q, since we said Q is the Bowman's capsule, in the Bowman's capsule, we don't see any proteins, okay? We don't see any proteins. So that means that had to be the afferent, okay? And then, of course, in the Bowman's capsule, we'll see high concentration of other contents. Now, in the efferent, wouldn't find any urea there, okay? In the efferent, there's no urea, but in the afferent, there's urea, there are proteins. So that's the kind of logic you're going to use to arrive at that. All right, so I think let's proceed. Yeah, someone is saying afferent is bringing blood for filtration, so it has all the components. That's a correct observation. Okay, I usually say afferent is the A for arrival or a later, then efferent for E for exit or a twala. So that way you can remember. Of course, afferent is always bigger than the efferent, although that information at this point does not help much yet. So let's go to the other questions. Which component of the urine shows the greatest percentage increase in concentration? Okay, I have the other questions elsewhere. So this is our the nephron, what it would look like. So where I have put the cursor, this is the afferent arteriole, like we mentioned. It is a larger vessel, and then it forms a dense capillary of blood, a denser network of blood capillaries, what we call the glomerulus. Okay, and then it exits, the blood exits it through the afferent before it later on joins another dense network of blood vessels, which we call the peritubular capillaries. Peri meaning around, tubular meaning the tubes. So it's a network of blood capillaries that are surrounding the tubules. The tubules meaning the tubes that are coming off from the Bowman's capsule. So when there's filtration inside here, the filtrate, which we call the glomerular filtrate, will enter through this white tube that is twisted or that is highly coiled. So because it is highly coiled, we say it is convoluted. The word convoluted means something which is highly coiled. And of course, it's called proximal because it is near the Bowman's capsule. Proximal meaning near. So it twists, of course, there are reasons why it is highly twisted, providing a very large surface area to allow for maximum reabsorption. Because when the filtrate, because when the filtrate is passing through this proximal convoluted tubule, remember it is surrounded by a network of the peritubular capillaries. So the contents in the filtrate are being taken back into this network of peritubular capillaries. And that's what we call selective reabsorption. So, of course, everything is recovered or reabsorbed. So what isn't recovered or reabsorbed continues down into the loop of Henle, as you can see here. Okay, but you notice that even in the loop of Henle, there's still a network of these peritubular capillaries, which continuously recover some of the useful contents in the filtrate. So all the way up here until we reach the distal convoluted tubule. Okay, it's called distal because it is farthest or it's very far away from the Bowman's capsule. So even here we have other reabsorptions taking place. Galaxy A2, can you tell us what you want to tell us?
Yes, I was asking, I want you to explain more about the descending and the ascending limb.
The ascending and the descending loop of Henle. Well, at our level at S4, we don't emphasize what happens in each of those loops. But what we need to know is that in the loop of Henle, there is reabsorption of water. So animals that have long loops of Henle tend to reabsorb much water than those that have shorter loops of Henle. And animals with long loop of Henle are those that are found in dry or arid environments. And then those with short ones or non-existent ones are those that are found in places where there's an abundance of water. So that is what I can say for now. But at A level, we shall tell you what exactly happens, how there is a secretion of sodium ions, and then how they cause water reabsorption and so on. But right now, for our level, we don't need to know those details. All we need to appreciate is that the loop of Henle is a site where water is absorbed back into the bloodstream together with the distal convoluted tubule, and the collecting ducts. All right. So I hope as we go through the next questions, we can bear this picture in our minds, okay? So that we can be able to answer the questions satisfactorily. Yeah, so these are the values we got for the earlier calculation. Where possible, you can take screenshots, but this information is being recorded and you can possibly access it at a later stage. So these are our answers, P, Q, and R, the afferent blood vessel or afferent arteriole, then Q, the Bowman's capsule or the capsular space, then R are the collecting ducts. Because in the collecting ducts, by that time, we are talking about urine, okay? We're talking about urine because all the other things have been removed, okay? Now, here are some questions that I want us to also attempt. We have a question which goes as follows. Which component of urine shows the greatest percentage increase in concentration compared to the blood plasma from part Q? Okay, let's go back and look at our table. Which component has the greatest increase in concentration? So if we look at our table, which component has the greatest increase in concentration? Seeing some, let's look at the chart. Someone wants to screenshot the other thing. That thing, you can get it on the net. I don't want us to take too much time. But let's see the people's answers. Someone, Camillus is saying water. What do the rest of us say? Galaxy A02, what do you have to say? Which thing had the highest increase in concentration? Galaxy?
Glucose. Glucose.
People are saying glucose.
Water.
Someone saying water. Master care?
Water.
Water?
Water. Yeah.
Not yet getting the answer I want. Rona?
Urea.
I'm sorry?
Urea.
Urea. So it is urea.
This is my class.
Urea. So it's actually urea. Now, why would there be a high increase in the amount of urea as opposed to all the others? Why does urea have the greatest increase? Let's have...
Yes, master cares.
Master cares?
This is because urea, it's not needed in the body, so it is excreted in the
Why isn't urea needed in the body? I think you need to tell us a little more. Can we be very courteous? If someone gives an answer, we should respectfully listen to it rather than laugh. Those who are laughing, I'm going to exit you the room because we need to know how to behave when we are having such sessions. Okay, let's have someone who has not said much yet. Cinderella?
Teacher, urea is not reabsorbed back into the body.
Why? That is true. but why isn't it reabsorbed back into the body just like others? Tracy?
Because it's a waste.
Urea is a waste product of the body which is toxic, so it is not reabsorbed by the body at any point. Therefore, it has the greatest increase in concentration of the urine, I think.
The answer, yeah.
Yes, so to start with, like she has said, urea is a toxic waste product. So there's no need for it to be reabsorbed. Now, when other things are being reabsorbed, urea's concentration increases instead. It's like when you're cooking food and you're cooking, say, meat stew, and then the water starts drying or evaporating. It means that the salt, which was originally okay for consumption, becomes very salty. It becomes very salty because the water has evaporated. So when you keep removing all the other components, it means that the concentration of urea goes on increasing instead. And like everyone has said, urea is toxic, so it should not be reabsorbed. And so because other things are being reabsorbed, so the concentration of urea will increase. Okay, if you're not talking, kindly mute. Jocelyn, Ivy, Harriet, please mute. Mute your mics until you are picked on to talk. Okay, Galaxy, what do you have to say? I'm seeing your hand is up.
Yes, Master. Can you pardon on the explanation why we have mentioned urea?
We said that urea is toxic. Remember, it's a waste product of metabolism. So that in itself makes it toxic. So there's no need for it to be reabsorbed. So we don't see anywhere where it is reabsorbed. So while the other components which are not toxic are reabsorbed, that in itself causes urea's concentration to increase. Okay? Daniela?
Teacher, I have a small question. In our paper, we had 3 marks for that, but I gave the same answer that I've given you right now, and I have 2 marks. So what was the other missing marking point?
So you have to tell us that the rest of the substances are being absorbed, so their concentration reduces, while the concentration of urea increases since it's not being absorbed. And why it's not being reabsorbed is because it is toxic. Is that clear now?
The other marking point was the others are not reabsorbed. Like that
No,
was what I was missing.
others are reabsorbed. When reabsorb something, the concentration of that thing reduces.
Oh, okay. Thank you.
Yes. All right. Okay, let's carry on. So here we are. I think I've elaborated on that. Then explain why protein in the glomerular filtrate is at 0.0. Masters?
Yes. The proteins have bigger molecules. So they can't be filtered out in the glomerular filtrate.
So the proteins are large in size, so they cannot be filtered out. That's correct. Do others have different answers? Jocelyn?
What if I say that proteins are insoluble?
No, it's not just about being insoluble. It's about the size. Okay? Proteins have very high molecular masses, and that makes them big. and they are unable to pass through the glomerular filtrate. Think about the glomerula, Think about the glomerula as like a filter. When you're making passion fruit juice, you often use a sieve, and the seeds cannot go through the sieve because they are big in size. Benjamin.
Yes, please, teacher.
Yes, your hand is up.
Oh, teacher, I was just saying that the proteins they are very large in size and cannot pass through the small pores of the glomerulus.
Yes, that's right. Okay, so I think we are all agreeing on the same. Then explain why the other components besides proteins are found in the glomerular filtrate. So we see in the glomerular filtrate that there were other components apart from proteins. How come for them they are able to go through? Why weren't they retained just like the proteins were retained? Why were they not retained? Galaxy?
Yes, as we have said that others have smaller particles, so the smaller particles allow them to pass through.
To pass through what? I think your answer needs to be complete, like you were writing it, okay? Let's give complete answers. Let's not leave the answers in our heads.
Okay, to pass through the proximal convoluted tubule.
It's not about passing through the proximal convoluted tubule. Where are they trying to pass through? Jocelyn. Okay, let's have Master.
I think they were able to pass through the glomerulus, This is because the substances were smaller compared to the pores of the glomerulus, so they were able to be filtered out.
Okay. Is it just because of the size? Why else are they able to go through the glomerulus? Is it just about the size?
They are also soluble.
Is it about the solubility? Certainly not.
It has a wider lumen.
Jocelyn, I'm only picking those who are raising their hands.
How about the walls of the glomerulus are permeable to them?
It's about the pore spaces.
Yes.
Pore spaces. But what happens in the glomerulus is what I want us to talk about. What takes place in the glomerulus that causes the production of glomerular filtrate? What happens there? Camillus?
Ultrafiltration.
Is ultrafiltration. Cementa, what is ultrafiltration?
Hey, mute.
Cementa, we are getting feedback from your ends. Okay. Joseph Atwesiga. Okay, let's hear from Peaches.
I think ultrafiltration is what occurs in the glomerulus when substance is filtered out into the capsular area. Something like that.
Yes, it's something like that, but you can give me a little more. Samsung Galaxy S7. Samsung Galaxy S7.
Ultrafiltration. This is the filtration. Are you getting me?
Loud and clear.
is the filtration of smaller molecules from blood in glomerulus under high pressure.
Okay. Yeah, so this filtration is taking place under high pressure, what we call hydrostatic pressure. Without the hydrostatic pressure, it means that the process would be extremely slow and would have very little filtration taking place. So we'll have very little filtrate, and that would not be sufficient to sustain the life of the organism. It means that the waste products will take longer to be extracted from the blood. Okay. And of course, what causes this pressure to build up in the glomerulus? Who can tell us what causes the pressure to build up from the glomerulus? Master Cares?
The pressure is created by the action of the heart, and it is also created by the diameter of the afferent arteriole being too wider than the afferent arteriole.
Okay. Yes, so the differences in the diameter of the afferent arteriole and the efferent arteriole contributes to the buildup in the pressure, whereby the afferent arteriole has a wider diameter or wider lumen than the efferent arteriole. If you want to appreciate what I'm talking about, open a tap and put your finger in the tap and see how water comes out of the tap. You'll notice that when you put your finger and partially close the tap, the water comes out at extremely high pressure. But if you remove your finger, the pressure of the water reduces. Yeah, so that is basically also what happens during ultrafiltration. So let's move on. So that is our answer. Of course, it's just summarized, but with the main points being captured. All right. Let's move on to the next question. You can quickly take screenshots if you may. Let's see what's in the chat. Master's Care, please. Master's Care. Your hand is up. Okay, I think they forgot their hand up. Let's carry on. Okay, now suggest with a reason the name of the health condition of the person from whom the results in the table were obtained. If we can go back, we look at these results. What kind of health situation does this person experience? You look at this table. Is this person normal? Are they healthy? And if they are not healthy, what condition would we say they are experiencing? And you give us a reason why. What condition are they experiencing? Benjamin?
The person from whom the results we got is suffering from diabetes mellitus.
Diabetes mellitus, that's correct.
And this is because of the presence of glucose in their blood, in their urine.
There's presence of glucose in the urine, not in their blood. We need to be careful. We can make that mistake and they end up missing. Jessica, it's not diabetes insipidus. What's the difference between diabetes insipidus and diabetes mellitus? Who can help us appreciate that difference? Master's care?
Diabetes insipidus is a condition where, and that somebody's blood, urine, there is large amount of water when that urine is dilute. But for diabetes mellitus is when there is glucose in that person's urine.
Okay, thank you. So if we carried out a Benedict's test on both people, whose result will give us a positive Benedict's test? Camillus?
The test for that one was glucose. Like that one was diabetes mellitus.
Okay, that's true. So you can take your own blood sugar test. You know, we are not supposed to have glucose in our blood, in our urine. So if you were to do a Benedict's test and then it turns out to be green, it means you have trace amounts of glucose in your urine. Turns out to be yellow, it means you have moderate. And turns out to be orange or brown, it means that you have a lot of it. And that should be an indication for you to go and get medical help. What could be the symptoms of someone who has diabetes mellitus? What are the observable symptoms? The person is dehydrated. They are thirsty, you mean? What else? Cinderella?
Obesity.
Obesity, not quite. There are people who have diabetes mellitus, but they are very thin. So it's not about the obesity. Rona?
Loss of body weight.
Yeah, sometimes they lose weight because they are losing too much sugars in the urine. What else, Vanessa?
Their wounds their wounds take long to heal.
Okay, they take long to heal. Shiberu?
Master, what about frequent urination?
Yeah, they tend to also want to urinate a lot. That's where there's a similarity between insipidus and diabetes mellitus. But the other thing is they get hungry very often. Okay.
What
so
of
they
blood vision?
Yes, and they have blood vision. They can't see very well because their blood vessels in the eyes are affected. Anyway, let's carry on. That was just a by the way question. So we have done that. Suggest with a reason how the amount of water in urine is affected when a person eats salty food. There are times when we eat salty food. How would that affect the amount of water in our urine? I want to see some new faces. YK Rose, please put up your hand. Others, we are getting feedback. Tracy?
I think the amount of water in the urine would reduce because when the salt levels go high, the pituitary gland would have to stimulate the production of ADH so as to reabsorb more water, so as to fix the salt levels in the body, something of that sort, I think.
Okay, can we have someone add on what she probably has left out? You're on the right track, but I feel like you've left out some important information. Rona? Okay, let's hear from Master Cares. Master Cares?
When
Okay,
a
let's
person
go to...
eats, when a person eats salty foods, the amount of water reduces. This is because increase in the salt concentration reduces water, reduces the percentage of water.
Reduces water. Yeah, water concentration. Yes.
Okay, thank you, Master Cares. Let's hear from Vanessa. Then we shall have Anita.
At the loops, some water and some salt are reabsorbed into the bloodstream.
Thank you for your contribution, Anita.
So when blood is concentrated and it passes over the hypothalamus, it causes the nephrons to reabsorb just enough water. So this will cause the urine to be concentrated and little.
Okay, you've added something new, the hypothalamus, which nobody else had said. Lindsay? Lindsay?
Teacher, I think that since the salt concentrations have increased, the body needs more water, like to lower the salt concentrations, like to bring it back to normal, to prevent bursting of the cells. Hence, more water is reabsorbed from the urinary system.
Okay, all have the ideas, but we are not going to earn all the marks if we present our points that way. We need to be very systematic. Okay, so first of all, there must be a way of the body detecting the unusual high concentration of salt in the bloodstream. So that's where the hypothalamus comes in, okay? So the hypothalamus is always detecting any changes in water concentration or salt concentration in the bloodstream, okay? So when it detects that, then it's going to cause the posterior pituitary lobe to secrete more ADH. And of course, ADH is anti-diuretic hormone. Anti means against, diuretic or means diuresis. Diuresis is like dilution. So it's a hormone that works against dilute urine. It's a hormone secreted to stop you from producing dilute urine. So the ADH hormone, also called vasopressin hormone, it has two names. The moment its concentration is high in the bloodstream, it will stimulate the kidney tubules, the collecting ducts and the distal convoluted tubule to become more permeable to water. So it means that those peritubular capillaries are going to now absorb more water back into the bloodstream. And that means that now the water in the glomerular filtrate reduces, and so we can produce concentrated urine. So my friend from Masters was struggling to talk about the osmotic pressure. So the osmotic pressure of blood increases when there is a high salt concentration. Now, if you have taken a lot of drinks, maybe you've had a party and there's a lot of free drinks around, it means that certainly there's going to be too much water in your bloodstream. And so the hypothalamus will detect the low osmotic pressure. And therefore, it's not going to stimulate the posterior pituitary gland, lobe of the pituitary gland. So it means that when the glomerular filtrate is formed, it will be very dilute, and the walls of of the distal convoluted tubule and the collecting ducts will be impermeable to the water. So it means that all the urine formed will be very dilute. So most of what you pass out will be dilute urine. It will be tending towards colorless because you have taken plenty of drinks. Then how would the quantity of urine be affected if a person lost a lot of blood after an accident? Imagine someone has broken a leg or broken a body part, and then so much blood is lost. How would the quantity of the urine be affected, and why would it be affected as thus? So we have a master cares.
When a person gets an accident and loses more blood, the quantity of urine will decrease. This is because losing more blood will lead to less blood going to the glomerulus. So less water will be filtered out. I mean, less urine will be filtered out.
Okay, to some extent you have answered. The last bit is where we have not got the answer very well. Benjamin.
Yeah, when a person is involved in an accident, the amount of urine produced will decrease. This is because since urine is a combination like of excess salts and excess water, there won't be any excess water or any excess salts since the person has been involved in the accident and has lost a lot of blood. And that blood may consist of that water that would have been taken out as the urine. So the urine amount decreases.
Not quite. It's true that urine reduces. Daniela?
I personally think the amount of urine would decrease because blood is a medium through which the waste products like urea and water are transported, and therefore the amount of blood being taken to the kidneys would reduce since the person has lost blood. Yeah.
Yes, when it reduces, then what? There's that thing you're not talking about. When the amount of blood going to the kidney reduces, then what? How does that cause a reduction in urine? Okay, this is what I had to say. So the urine quantity reduces because excess bleeding will reduce the blood pressure. So when the blood pressure goes down, it means that in the glomerulus, there will be less hydrostatic pressure, and therefore there will be a lower filtration rate. And when we have a lower filtration rate, it means less urine is going to be formed. Okay. And when less urine is formed, of course, then the amount of urine produced is going to be low. So this filtration rate and hydrostatic pressure is what we have been missing out in answer. The next time I'm going to exit some of these offenders from our room because they are disrupting us. Okay, so that was one of the questions. I have another question that I want us to look at. It's a very simple one. I hope so. A graph was plotted to show the time taken for the enzyme to digest protein at each temperature. So we're dealing with pepsin and egg albumin. So can we quickly answer this question? What's the optimum temperature for this enzyme? Akram? Akram? I think Akram is not with us. Master Cares? Camillus?
(40°C).
Camilla says 40. Do the rest of us agree that it is 40? Let me call out some of these silent people in our room. Luther, can you give us the answer? Okay, let's
It's
hear
35.
from Vanessa.
20°C.
90 degrees?
I say 20.
20 degrees? Other people don't agree. Rachel Oswald?
40°C.
So there are two people are saying 40. So actually, Peter Chiberu?
Master, for me, I'm saying 10.
10? Okay,
Yes.
now this question is really interesting. Okay,
70.
Let's have it's written as 70. Let's have Camillus try to explain to us why she chose that answer. I'm seeing people saying 35, coach.
I chose it because.
So coach will tell us why he or she thinks it's 35.
I think it's 40 because when I look on the graph, at 0°C, it takes more time to work on the egg albumin, but on 40, it takes like 5 minutes. It takes 5 minutes, which is less time to work on the egg albumin, which makes it the optimum temperature for the enzyme to work.
Okay, Koji, thank you. That is good. Koji, you had a different answer. Coach, you have unmuted. Please talk.
And actually, like.
Okay, Mungufeni.
Yeah, I think it's 30 to 40 because in that range, the rate at which it was being the experiment was going on well was constant from 30°C to
Okay, thank you. So the answer is between 30 and 40 because that is where the rate of the time taken is lowest. Okay, but some people have said 35. So 35 would have been so if we had something like this. Okay, but we don't have such a thing. So because we don't have such a thing, we can't say 35. I know some people have tried to calculate the average between 30 and 40, which is a 35. But here we have a range of between 30 and and 40. Now, some of us are getting a bit confused. This is a unique graph where we are seeing how much time it takes for the reaction to be completed. So with this question, we got Egalmin. Egalmin is usually a turbid solution. When we put the suitable enzyme, which is pepsin, it is going to make the solution turn from turbid to colorless. And then we know there has been complete digestion. But if the solution remains turbid or it takes a longer time for it to turn colorless, it means that the process of digestion is slower. And the process of digestion is slow if, one, the temperature is not the correct temperature, so that very few enzymes are activated, or other factors that affect enzyme activity are affecting that process. Of course, those other factors, we are going to answer them when we look at question number 4. I don't want to talk about them yet. Okay, so if there are other factors affecting enzyme activity, then they can cause the reaction to take longer to be completed. So here, the shortest time taken shows us the optimum temperature, and the longest time taken shows us the temperature that is unsuitable. We can also have here pH. We could have a similar graph, but instead of temperature, we substitute it with pH. So if we substitute it with pH, now it would mean, if we had something like here, pH, it would give us the same results. Like some of you have been doing practicals where you are working with, say, hydrogen peroxide, and then you have an acidic medium, and then here you have neutral medium, and then I have alkaline medium. And then we see the time taken for the bubbles to be formed. You find that where the pH is most suitable, the bubbles will form very quickly within a very short time. And where the pH is unsuitable, it may take longer for those bubbles to form, or sometimes the bubbles are even fewer. Okay, the next question is, let's give an account for the time taken to digest egg almond at 70 degrees. Why at 70 degrees do we have such a time taken? Why is it taking this long? As someone will tell us why it also takes this long at 0 degrees. So Camilas, tell us for 70 degrees. Benon, tell us for 0 degrees. Why the time taken is that long?
At 70 degrees Celsius, it takes more time because high temperatures denature enzymes, and so the enzyme reaction, the action on the egg albumin will be slow, and it will take more time.
Thank you. So most of the enzymes are denatured at that time, okay, at that temperature. Benon, what about at 0?
At 0 degrees Celsius, the temperature when it's in an inactive form, it won't be able to.
We have lost Benon. Master Cares.
Yes, when temperature is at 0, the enzymes are inactive and of which enzymes work best at optimum temperature. That makes it to lower to 0.
Okay, thank you.
Take more time.
So at low temperature, enzymes are inactivated. Remember from what we call the kinetic theory of matter, that when you raise the temperature, molecules increase the speed of movement. And so it means that there will be more interaction between the enzyme and the substrate, forming more products. But when you lower the temperature, it means that very few enzymes are going to be interacting with the substrate. So there will be fewer products formed. So it will take long for the enzyme to cause the reaction to proceed to completion. So that's why we say that at low temperatures, enzymes are inactive or they're inactivated because they have low kinetic energy to collide with the substrate. But at high temperatures, we see that there's too much kinetic energy that even causes the molecule itself to become denatured. It breaks down. The active site becomes distorted, that it can no longer even interact properly with the substrate. It does not bind with the substrate because the active site has been distorted. So that's why we say that enzyme has been denatured. Okay, Harriet, we are hearing you. Don't know if you intended us to hear what you're saying. All right. Now, with the reason, name the form in which pepsin is secreted. What's the form in which pepsin is secreted in your body? Pepsin, I'm seeing someone here saying pepsin is secreted in an inactive form called pepsinogen. Prevent the digestion of the stomach walls, which are protein in nature. That's wonderful. Okay. Yeah, so it's secreted in an inactive form called the pepsinogen. And that is on purpose. Okay. I think we can go on to the next. State 3 other factors that affect enzyme control reactions. Other factors. I think I saw some hands somewhere. Jocelyn?
Effect of
give me one factor. Others will give us the rest.
Effect of enzyme concentration on the enzyme activity.
Tell us a bit about that one.
I'm not certain.
Okay, who can build on Jocelyn's answer? How does enzyme concentration affect enzyme reaction?
It is because, like, the more concentrated the enzyme is, the faster the work of the enzyme on the substrate.
The faster the rate of reaction.
Yeah.
Of course, that will happen up to a certain level where the rate of reaction becomes constant. We are going to get something like this. Let me annotate. So you get something like this. It flattens later. Why does increase in enzyme concentration? Actually, it's substrate concentration that causes the flattening. I've given you the answer already. So if we have substrate concentration here increasing, then later on the rate of reaction flattens. Why?
It is because, like, the enzyme works on the active sites of the substrate, and then when they are over, the reaction will be constant.
Okay, so what happens is that all the active sites are saturated or they are occupied by substrate, okay? Because the enzymes are of a limited quantity. So it means even if you increase substrate concentration, the rate of reaction cannot go further. So for you to take it further, you'd probably need to you probably need to increase the enzyme concentration. Then you'll have something like this. Okay. It's like if you go to the canteen and you have 3 serving points, and yet you are 2 students. It means that 1 serving point is free. If you become 3 students, all the serving points are occupied. If you become 10, only 3 of you can be served while the rest of the students wait. So if you want all the students to be served quickly, then you must increase the number of people who are serving. Okay? So basically that is the whole essence. What other factors can affect the rate of enzyme reaction? Apart from substrate concentration and enzyme concentration, what other factors?
Surface area.
Please, let's not talk out of turn. If you want to say something, you raise your hand. Let's behave in a civil way. Jocelyn?
Effect of heat on enzyme activity.
That one was already mentioned in the question, temperature. So we can't use it as an answer there. Let's have YK Rose. YK Rose. Okay, let's hear from Rona.
Presence of inhabitants.
We call them inhabitants. What's the right word?
Inhibitors.
They are called inhibitors. Okay, let's hear from Mugide Mary.
Presence of medium of the pH.
Okay, pH of the medium, you mean? pH of the medium. Other factors. Master care. Benjamin.
Is it also okay to mention enzyme cofactors?
Yes, enzyme cofactors. Okay, so those are some of the other factors that could affect. Now, number 5, there are times when we have fresh milk, and if we forget it on the table, say overnight, we find when it is spoiled in the morning. But if we put it in the fridge, it doesn't get spoiled. How do we explain this? Oppo F15. Oppo F15.
Excuse me, teacher. I was asking just what you've previously talked about. What are the inhibitors and how do they affect enzyme concentration? I mean, enzyme activity, sorry.
Inhibitors are substances that can either occupy the active site, stopping the substrate from occupying the active site, and therefore no reaction takes place. Or they can occupy another part of the enzyme, still stopping the substrate from binding with the enzyme. So anything that stops the substrate from binding with the enzyme is called an inhibitor. It's like if you want to read books and there's someone who is creating an environment unfavorable for you to read, that person becomes an inhibitor to your success. Okay? So in physiology or in our bodies, there are substances that work as inhibitors. Some of them can be poisons. And that's why people take poison and die, because those inhibitors stop normal functioning of enzymes. And when someone's enzyme doesn't function well, it means that many metabolic reactions will not happen, and that could cause the person to even die. I hope that is clear. So let's have the answer to number 5. Vanessa?
So I just wanted to confirm whether my answer is correct. What if I say that the inhibitors block the active site of the enzyme, preventing the food substrate from being worked upon? Is it correct or I'm wrong?
Your answer is correct, except for the word food. Because enzymes are not only about food. So you just said they prevent the substrate. The substrate can be a food molecule, or it can be any other molecule, okay?
Okay, sir.
Yeah. Let's have some for the answer number 5. Number 5. Camillus.
Like when the unboiled milk is on that table, like it's the enzymes in the milk will be given optimum temperature so as to work on the enzyme to make it to work on the milk so that it gets spoiled. But when it's put in the fridge, the low temperatures in the fridge will inactivate the enzymes so the milk won't get spoiled.
A nice attempt. Let's have Peaches. Peaches has run away. Master Cares.
Unboiled milk left on the table easily gets spoiled. This is because there is optimum temperature for the enzyme activity. Hence, the milk gets spoiled easily. While in the fridge, there is no suitable temperature, so hence the enzymes are inactive. Hence, there is no enzyme activity. Thus, the milk.
Okay, thank you, Master Cares. There's something that we are all seeming to miss, just one little thing. Maybe YK Rose will tell us. YK Rose? Okay, let me pose a question. Why does milk go bad? What causes milk to go bad? Chiveru, Peter?
I think it's fermentation. I mean, like bacteria fermentation.
Wonderful. So there are bacteria that ferment or spoil the milk. Why do these bacteria spoil the milk? What happens with the bacteria that causes the milk to go bad? Yes, Ikeros? All right, so Oppo A15.
Teacher, I think the bacteria breaks down the soluble caseinogen into insoluble casein, hence the milk gets spoiled.
Nice attempt. So we know that bacteria, there are some bacteria we call the lactobacilli. They feed on the milk sugars. You've heard of the lactose. So as they are feeding on the lactose, they do so by fermentation, or they produce other products which are acids. Those acids are those which cause the milk to get spoiled. So it's basically the presence of the bacteria as they are breaking down the milk, like one of you has said. So if you provide optimum conditions, like what happens at room temperature, the bacteria will thrive. They are going to multiply and they are going to cause that process to speed up and the milk will go bad. But if you put in the milk in the fridge, it means that those bacteria are going to be inactive. Okay? And when they're inactive, it means even their enzymes are not able to work. So it's about the inactivity of the enzymes in the bacteria, okay, that causes the milk to survive. So I wanted you to relate the bacteria and the fact that the bacteria also have enzymes. So the enzymes in the bacteria are used to break down the milk into products that cause the milk to go bad. So they produce those acids, okay? And those acids are what cause the milk to go bad. So that is something of practical application in our everyday life. So if you don't have a fridge, what do you do to keep your milk from getting spoiled? Maybe you don't own a fridge, or maybe the power has gone off. So what do you do? Do you give up the milk? Yes, Master Cares?
The milk. Boil the milk. You boil the
What did
milk.
you say?
You boil the milk.
You boil the milk. Why do you boil the milk?
Boil the milk.
What are you trying to achieve by boiling the milk? Sorry?
You denature the enzymes responsible for spoiling the milk.
You drink the milk.
Camilla, someone is saying drink the milk. So you boil the milk to kill the bacteria, okay? So that the milk doesn't get spoiled. Remember, bacteria are found everywhere around us, even in the air we breathe. So these bacteria are attracted to substances that are nutritious. Milk is very nutritious, so it attracts a lot of bacteria. Even meat is also nutritious. It attracts a lot of bacteria. That's why meat also gets spoiled very quickly. Let me pose another question. Why is it that when you salt meat, the salted meat doesn't get spoiled? What is the logic behind salting of meat? Why does salted meat last longer? Using the biology you know. Anita?
So when you add salt to the meat, the bacteria present, their cells shrink. So they won't be able to do anything when the cells have shrinked.
Okay, thank you, Anita. So adding salt will cause the bacteria to lose a lot of water by osmosis, so they will shrink and die, okay? That's why honey doesn't go bad, because honey is a very strong solution of sugar. So whatever bacteria falls in the honey will die. So honey can be kept for ages without it getting spoiled. All right, let's go to my last question that I had planned for us.
Excuse me, sir.
Yes, please.
Would you please pardon the answer?
Which answer to which question?
Why do we salt meat?
Yeah, when we salt the meat, we are causing any bacteria present on the meat to become dehydrated. Okay? To become dehydrated. So that's why we salt the meat. And when the bacteria are dehydrated, they cannot break down the meat into and cause it to get spoiled. All right, so are these questions or we proceed? Okay, let's go to the question that was supposed to be your take home. Maybe we can discuss it. Describe the events that take place in the mammalian heart that cause blood to enter the auricle and leave by the ventricle. What is this topic? What does this require from us? What is the topic? Someone says blood circulation, blood circulation. The topic is called transport. Blood circulation is a subtopic. Okay. So who can tell us what this question wants us to discuss? Oppo A15.
It wants us to discuss the cardiac cycle.
Okay, the cardiac cycle. Would you want to take us through the cardiac cycle? Anyone wants to take us through the cardiac cycle? Okay, so I'm going to show you an animation and then maybe it can spur us into discussing the cardiac cycle. So I want us to observe this, then we shall start our discussion.
This animation illustrates a single cardiac cycle, starting with the right side of the heart. In reality, of course, the two sides of the heart work together at the same time, but it is easier to understand if we look at them separately, because each side of the heart has a different function. The cardiac cycle begins as deoxygenated blood flows from the body into the right atrium via the superior vena cava and inferior vena cava. In the next phase of the cycle, the right atrium contracts, pumping blood into the right ventricle through the tricuspid valve. Once the right ventricle fills with blood, the tricuspid valve closes to prevent blood flowing back into the atrium. The right ventricle contracts, forcing the pulmonary valve to open as blood is pumped into the pulmonary artery. Blood then travels to the lungs and the pulmonary valve closes. At the same time, the right atrium relaxes, ready to accept the next intake of blood. In the lungs, the blood picks up oxygen, releases carbon dioxide. Oxygenated blood flows from the lungs into the left atrium via the pulmonary veins. The left atrium contracts to pump blood into the left ventricle through the mitral valve. Once the left ventricle has filled with blood, the mitral valve closes. Next, the left ventricle contracts, forcing the aortic valve to open, allowing blood to be pumped into the aorta, and from there around the body. The aortic valve closes to prevent blood flowing back into the left ventricle, and the left ventricle relaxes, ready to accept the next intake of blood. Cardiac cycle is now complete.
Okay, I hope we have followed very closely, and now we can be in position to answer the question. This question is not very specific to which part of the heart. It's very open. So how you answer it should be able to cater for both the left and the right hand side of the heart. So who is willing to explain to us what they have understood from the animation that we have just observed? Any volunteers? Yes, Peter?
Master, I'm trying.
Yeah, that's good. Then Vanessa will come after you.
Yeah, it starts from the right lung through the vena cava to the right atrium via the tricuspid valves, then to the right ventricle, then to the pulmonary artery, then to the lungs, from the lungs to the pulmonary veins, then to the left atrium via the bicuspid valves, to the left ventricle, and to the aorta, where it pumps to all body parts.
Thank you, Peter. You have given us the route taken by blood, but we also want you to talk about what causes the blood to move from one part of the heart to the other. The question wants from the time the blood enters the heart to the time when the blood exits the heart. What things are happening in there? Okay. Thank you for your contribution, because you've told us about the lungs, but those are not part of the answer. They are correct answers, but not for this particular question. Vanessa?
Just trying and in summary, like when the heart is full of blood, it will undergo cytocolic phase, whereby the heart muscles contract drumming. The heart blood, that is to say blood, moves out of the heart to the lungs, and the rest of the body via pulmonary artery and aorta respectively. And also when the pocket valves open, the closing to the produce of the sharp sound, which is heard as
Thank you, Vanessa. I want a very simple answer. What you're taking us through is quite complex. Let's keep it very simple. Cinderella, before you come, I want you to talk about how the blood enters the heart. Okay? Where does it enter from? The question is already telling you it enters at the auricle, or what we call the atrium. So what causes blood to enter the atrium or the auricle, and what causes it to enter the ventricle, and what causes it to leave the ventricle? That's why I want our answer to stop. Anything outside the heart, let's not talk about it, okay? Whether lungs or rest of the body parts, let's ignore that. Let's concentrate on what is happening in the heart. So Cinderella, you're next, then Tracy will come afterwards.
Okay, teacher. Blood enters the heart through the pulmonary vein and the vena cava. This occurs when the auricles and ventricles are relaxed so that the pressure is low. Then, when the auricles are filled with blood, the pressure is going to increase, and this high pressure forces the cuspid valves to open so that the blood can flow to the ventricle. The semilunar valves are going to close so that blood does not flow back to the where it came from. Therefore, when the blood flows to the ventricles become filled with blood. When they are filled with blood, they also contract, and this causes an increase in pressure. Therefore, the high pressure of blood in the ventricles causes the blood to flow through the pulmonary artery to other parts of the body.
Okay, that's a lot better. Although you missed out a few things, but that's a lot better. Okay, does anyone, there was Vanessa. Vanessa, you have run away. Okay, I will share with you what I had prepared for the same. And I want us to read it carefully so that we can. So in my answer here, I'm looking at the left and the right concurrently, like what Cinderella was trying to do. We have the vena cava for the right-hand side and the pulmonary vein from the left-hand side. So blood enters each half at the same time. So the vena cava and the pulmonary vein will relax. So when they relax, it means that the lumen becomes wider, okay? So it allows blood to flow from them into the atria, into the atria. So at that point, they will cause the atria to fill with blood. Now, when they are filled with blood, there's a tendency for this blood to want to go back from where it came from, because the atrioventricular valves, the ones which are connecting the atrium to the ventricles, are actually shut. So the blood is trying to go back because of the pressure, okay? And so when that happens, there will be some stoppage of that from happening. There are some valves that stop that from happening. So the muscular walls of the auricles will contract, causing the volume of the atria to reduce. Remember from physics, pressure is equal to force over area. So if the area or the volume is increased, the pressure reduces. If the volume reduces, then the pressure increases. So the volume is reducing due to the contraction, okay, of the atria. And when that contraction takes place, then the pressure of the blood in the atria will increase. So this pressure will force the valves, the tricuspid and bicuspid valves, what I've called the atrioventricular valves, because they are between the atria and the ventricles. So it causes those valves to open. So when the valves open, it means now blood is forced to flow only towards the ventricles. So when the blood enters into the ventricles, it will continue flowing there until the ventricles are now filled with blood, okay? So while the ventricles are filling with blood, the walls of the atria are relaxing, okay? And while they are relaxing now, the walls of the ventricles are beginning to contract. So when the walls of the ventricle are beginning to contract, the blood tries to go back to the atria because the pressure is less there. Now the atrioventricular valves, or the tricuspid and bicuspid valves, now will have to shut so that the blood does not go back to the atria, okay? And when that happens, the pressure also builds up sufficiently to cause it to flow through the pulmonary artery and the aorta. And when the blood tries to come backwards to the ventricles, then the semilunar valves will shut, preventing blood from flowing back into the heart, and then it is pumped away from the heart. So basically, that is what is expected of us. So you have to go systematically, okay, from the atria to the ventricle. Now remember, blood enters the atria because there is low pressure and a high volume. So blood flows from high pressure to low pressure. So if the atria at a low pressure, blood enters them. Excuse me. And then blood exits the atria into the ventricles because now the atria have a higher pressure than the ventricles. So blood flows from high pressure to low pressure in the ventricles. And then blood leaves the ventricles to the rest of the body because the pressure in the ventricles has increased due to the contraction of the walls of the ventricles. So I hope that is clear. If you remember the video I showed you, that was basically that was taking place. Let's go on to the last two parts of the question. Adaptations of the heart to their function. How is the heart adapted to its efficient functioning? Expect answers from us.
It has the cardiac muscles too.
Cardiac muscles, what is special about them? When we talk about adaptation, you must describe something and tell us how that description makes a thing function well. So if they are cardiac muscles, what is special about cardiac muscles that is not found in smooth muscles, that's not also found in skeletal muscles? What makes cardiac muscles so special that they must be part of the heart and make the heart function well? Chiberu? Okay, who can help him? Oppo 115?
The cardiac muscles are thick and tough to withstand the high pressure of blood into and out of the heart.
Okay, thank you. But the cardiac muscles, it's about not fatiguing or getting tired. You know, when you participate in any sporting activities, okay, it means after a while you get tired. Now, your cardiac muscles have been pumping blood in your heart from the time you were a little baby in your mother's womb up to this very moment. They have never gone on holiday. So we say that they do not fatigue, which is a very special thing about them. And someone in the chat has told us that the cardiac muscles are myogenic. Myogenic means that they do not get to contract as a result of outside influence. The central nervous system does not initiate their contraction. The contraction of other muscles is under the control of the central nervous system. These ones are self-starting muscles. They keep contracting and relaxing without any intervention, to the extent that if we cut out a heart out of an organism and put it in a solution that is isotonic, the heart continues to pump even when it's outside the body. Amazing. No other muscles are capable of that. So that's why we say that they are myogenic. Okay, other adaptations. I'm seeing someone telling us it has the atrioventricular valves, which prevent backflow of blood into the heart. So we can just say they have valves, if we are to use that point, that prevent blood from flowing back. Why shouldn't blood flow backwards? How important is that? Why is it such an important thing? What would happen if blood flowed backwards? As a biologist, you have to ask yourself such questions, okay? What if things happen otherwise? Is there any consequence? Is there any problem? So when blood flows, yes, Peter Chiberu?
Master, I'm trying.
Yes.
For me, I think it's because of it might cut the supply of oxygen to the different body parts like the lungs.
Okay, nice try. Well, we want blood to flow in only one direction, okay? If there are shortcuts and blood flows in other direction, it means that even maintaining the pressure of the blood will not be very possible if some of it is being channeled into other places or if it is flowing backwards. So the valves ensure unidirectional flow of blood, which is important to maintain the blood pressure. Okay, what other adaptations, apart from the valves and the cardiac muscles, what other adaptations are there? Jocelyn?
The heart has veins which are thick to transport blood at low pressure.
Not quite. I'm talking about the heart itself. The veins, remember, are bringing in blood. So the veins don't have any influence on the functioning of the blood. Daniela?
Okay.
The heart has pericardial fluid between its pericardium so as to prevent friction.
Okay, that's true. The pericardium has a pericardial fluid, prevents friction. What else, Anita?
There's a masticular septum which prevents mix-up of oxygenated blood and deoxygenated blood.
Okay, that's also wonderful. Okay, any other?
Why is one no?
Chiveru?
The heart has the septum to separate oxygenated from deoxygenated blood.
Yeah, I think that has been mentioned by Anita. Okay, so here they are, the very answers you have given me. There's something that we have forgotten, the last one, that the left ventricular walls are very thick. So to generate energy that creates a high blood pressure, to pump blood to all the parts of the body. Okay. Daniela, your hand is up.
Teacher, could you give it the heart's location as an adaptation for its functioning, because for something to function properly it has to be protected. So could you say the heart is protected by the rib cage or something? I don't know.
Now that is not an adaptation for the heart. Okay, the rib cage, yes, true, protects the heart from injury, but that's not an adaptation of the heart. You get it? That's now an adaptation of the skeleton, perhaps. Okay, and the last question, and we call it a day, is what happens to the heart rate of a person who is scared after seeing a fierce dog? Explain the effect on the heart rate. So I want you to tell me the chronological events, the step-by-step events that happen that result in someone responding when they see a fierce dog. So this question covers transport and some other topic. Coordination. So let's have someone answer. Peter?
I think when a person sees a fearless dog, the hypothalamus responds by secreting like adrenaline responsible for emergency actions, which results into the faster rate of blood pumping.
Thank you, Peter. Benjamin?
Yes, teacher, when a person sees a fierce dog, the adrenal glands are stimulated to produce adrenaline, which increases the metabolic rate and thus increasing on the heart rate. So increasing on how the heart beats, increasing on the person's heart rate.
Okay, you've added something good. So the adrenaline is not secreted by the hypothalamus. The adrenaline is secreted by adrenal glands. You can hear the name, adrenaline, adrenal glands. So the adrenal glands, remember, are found on top of the kidneys. So the adrenal glands secrete adrenaline. Of course, the adrenal glands have two parts, the adrenal medulla and the adrenal cortex. So adrenal medulla is one which secretes the adrenaline. So the adrenaline, being a hormone, is directly released into the bloodstream, and its effects, hormones have what we call target organs. The target organs in this case are the heart and the lungs, as well as the muscles. Okay, so it's going to cause the heart to increase its heartbeat or heart rate. Okay, and then it's going to cause the blood vessels to other organs, maybe to the stomach, maybe to the bladder, to constrict. That's why when you are scared, you feel like your stomach has been tied into knots. You feel something funny in your stomach when you are scared. Okay? So the blood should not go to those other organs because they are not important at this point. So there's less flow of blood to those other organs, but instead the blood flow to the limbs and to the heart will increase, okay? And when it increases, it means that now the limbs are going to get sufficient oxygen so that the muscles can contract in order to make you either run as fast as you can or to fight that dog. Remember, adrenaline is called the fight or flight, hormone. So you either fight the situation or you fly away, as in you take off from the situation. So basically, that is how your heart rate would change because of seeing a fierce dog. Of course, the last question was about what other factors can affect your heartbeat or heart rate apart from fear. What other factors can cause your heart rate to increase or to decrease? So that's the very last question. Let's have that and then we can call it a day. Why, Rose? Okay, Daniela?
Hormones.
Hormones. Shalom?
Excitement.
Excitement. Of course, you'll be able to explain each one of those. Another one? Exercise. Sickness.
Body size.
Body what?
Size.
Body size, true. Okay. All right, so go and think about how to explain those, and then we shall meet next time. So it has been nice interacting with you. I started with 50 at some point. I think that was the highest. And now we are 37. And because God has been good to us, for this medication and alcohol, that's correct, has been good to us. Our network has been very stable. I'm going to request one of us to thank God for this session and pray that people can also be able to join us next time, but also guide us as we are revising. So I think Vanessa is going to pray for us, and then we shall call it a day.
In the name of the Father, and the Son, and the Holy Spirit, kind and merciful Father, we thank you for the protection which you have provided to us, the teaching for our teacher has taught us. So Lord, please help us, let it stick into our minds and make it in practice. Let's utilize it and make it confirmed. In Jesus' name I prayed and believed.
Amen. Thank you.
Amen.
So we can go and help our parents and be good people.
Excuse me, teacher.
Yes, please.
Teacher, I'm humbly requesting you to send the work to the WhatsApp group, the work that we have done today.
The recording is on. I don't know, the admins will find a way of sharing it, because I'm not part of the group, the WhatsApp group. I can't share anything.
Excuse me.